Hey there, my name is Nathan Lapierre. I'm a PhD student in computer science at UCLA, and I'm going to be talking to you about our method Metalign for metagenomic profiling. So there's a couple terms to define here. Metagenomics is whole genome shotgun sequencing of microbes recovered directly from their host environment, things like bacteria and viruses. And profiling is predicting the organisms present in the sample and their relative abundances. So you would output something like E. coli is 30% of the sample, S. aureus is 12% of the sample, etc. And this is usually done using a reference database. So what the most sort of intuitive way you can think of doing this is by just aligning reads to the reference database using something like BLAST. And many people still do this because this approach is considered highly accurate. It's been demonstrated to be highly accurate. But the problem is it can be computationally infeasible. So these reference databases are often hundreds of gigabytes in size. You can have tens of millions of sequence reads, so it can take months to blast the reads against a reference database. And various other heuristic approaches have been developed, but they're not as accurate. So we wanted to ask the question, can we recover the accuracy of an alignment-based approach while making it computationally feasible? And our solution to doing that is called Metalign, which we call efficient alignment-based profiling for that reason. And so our intuition is that the main bottleneck here is these giant reference database sizes, but most of the genomes in that reference database can quickly be ruled out as not being in the sample because they share almost no genomic sequence with the reads in the sample. So can we have a quick, efficient pre-filtering stage to quickly eliminate a lot of the organisms and then perform alignment on a small subset database? So that's the Metalign approach. We do this pre-filtering, then alignment, and then we deal with multi-aligned reads, which are reads aligned to multiple genomes, and compute this output profile. And so that pre-filtering stage, really the key step, is called containment min-hash, as mentioned in the title, SMASH for short. And so what it does is it estimates the containment index, which is basically the percent of k-mers in the genome that are thought to be in the reads. And this has been theoretically shown to be superior to the Jacquard index. So we test each genome, find its containment index, and then the genomes with a containment index above a certain cutoff threshold, we put in our subset database. And then we perform alignment against this subset database. And you can quickly actually rule out the vast majority of organisms that way. So we have a comprehensive database of all assemblies on NCBI, RefSeq, GenBank, and even some that aren't in GenBank. And this database is about 243 gigabytes compressed. In practice, we were able to subset this down to about half a gigabyte to 5 gigabytes, depending on the diversity of the sample, which makes alignment feasible. And we do Minimap too for the alignment, an efficient aligner. And then finally, as I mentioned, I'm not going to go into details. We process reads aligned to multiple genomes, which is critical for estimating the abundances accurately. And then we output the profile. And we show in the paper that doing this versus just running Minimap against the whole database obviously dramatically reduces runtime. It actually also dramatically reduces false positives because there's a lot less spurious alignments of reads to organisms that aren't in the sample. So that's the Metalign approach in a nutshell. And we compared against some of these other methods, most of which are some of the other heuristic-based approaches, some k-mer matching approaches, some marker gene approaches. And we use this critical assessment of metagenome interpretation. This was a big metagenomics benchmarking competition, had 8 data sets, highly diverse, a lot of novel strains simulated, so a lot of sequences that aren't in your reference databases. And so we compared against a number of other methods here. And here I'm showing on the genus level, averaged overall 8 data sets. And what immediately stands out is Metalign, MOTUS2, and Megan is another alignment-based approach, stand out in terms of precision and recall. And also an L1 error, which is a measure of abundance estimation error, which here I flipped and rescaled such that one is better, like the other metrics. So these 3 methods really stood out. The other methods, Bracken, Clark, Gotcha, MetaBin-G2, MetaFlan2, not MOTUS2, struggled in either recall or precision. Megan, which was highly accurate, another sort of alignment-based approach, however, was dramatically slower than the other methods. So summed over all 8 data sets, 50 million paired end reads each, Most methods took in the vicinity of 10 hours. Megan was a couple of orders of magnitude higher in terms of wall clock time. Clark was the highest in memory. Metalign was middle of the pack of both. Our goal is not to be the very fastest method, but to be reasonable and the most accurate. Megan's also accurate, but it took a lot longer. And MOTUs did really well in this data set and was reasonably efficient. We looked at another setting, which is a mock community, which is basically a community that's created in a lab and then sequenced. So you get the advantage of the noise of sequencing real data, but you know the ground truth, so you can actually measure your performance. So that's why mock communities are nice. We used this one by Peabody et al. And compared the methods on a variety of different abundance cutoff settings. So this means organisms with below this abundance in the sample were removed, which helps some methods like Bracken and Clark that generate a lot of low abundance false positives. And for any non-zero abundance cutoff setting, Metalign was the highest performing method. Interestingly, on this data, MOTUs too was generally the worst performing method. So these two very different settings, Metalign was the only method that performed well in accuracy and reasonable in runtime and memory on both of them. And we're able to do this by leveraging an alignment-based approach in combination with a highly comprehensive reference database. And the way we're able to stay efficient by doing that is by using this smash-based pre-filtering, which dramatically reduces the database size and makes runtime feasible. And the way we're able to use a really comprehensive reference database without exploding in memory is because Smash uses some lightweight K-mer sketch data structures. So it's also memory efficient. So those are the key takeaways. I'd like to thank my co-authors. I'd like to thank you for listening. We're on bioRxiv and GitHub. Thank you very much.